Hello and welcome to this webinar, Evaluation of Checkpoint Inhibitor Therapies using an MLR Assay. I'm Lou Rahman, Editor-in-Chief at DDW. Today's webinar will be presented by Kirsty McBain, Associate Scientist at Sartorius. Immune checkpoints are signalling molecules that control the synapse between T cells and antigen-presenting cells. Checkpoint inhibitor therapies target and inhibit the signals that switch off T cells, leading to an upregulation of T cell activation and tumour cell killing. The mixed lymphocyte reaction mimics the T cell and APC synapse by co-culturing immune cells from two individuals. MLR is used as a model to explore the effects of checkpoint inhibitor drugs in vitro, facilitating drug discovery processes and biological research applications. In this webinar, Kirsty will focus on applications of the iQue Advanced Flow Cytometry Platform. She supported a number of the Sartorius immuno-oncology campaigns and will use this webinar to discuss how the iQue Advanced Flow Cytometry Platform and associated reagent kits can be used to quantify T-cell response in MLR, the use of the iQue MLR assay to generate pharmacological outputs for checkpoint inhibitor-induced T-cell activation, the impact of checkpoint inhibitor therapeutics on inflammatory cytokine release, and she will also provide live answers to your questions. Before we start, a little bit about Kirsty. Kirsty is an associate scientist in the iQue Applications Group, part of the European-based bioanalytics at Sartorius, where she's involved in the research and development of novel applications. Kirsty's efforts are focused on the iQue3 Advanced Flow Cytometry Platform, where she's supported a number of the immuno-oncology campaigns that are used to exemplify how Sartorius technology can be applied to the drug discovery process. Kirsty obtained a master's degree in biochemistry at the University of Bristol before joining Sartorius in 2019. Kirsty's presentation will be followed by questions. Kirsty, over to you.
Today I'm going to be presenting this webinar focused primarily on investigating the effects of checkpoint inhibitor drugs, titled Evaluation of Checkpoint Inhibitor Therapies using a Mixed Lymphocyte Reaction Assay. So first on the agenda is a general overview of some of the uses of advanced high-throughput flow cytometry in biological applications. Then I'm going to give some more detail on the iQue3 platform, which is the advanced flow cytometer that was used to create the data displayed in this webinar. I'll then give an overview of the mixed lymphocyte reaction and how it can be used as an in vitro model to measure the effects of checkpoint inhibitor drugs. Finally, I'll cover two case studies that will exemplify the uses of the iQue MLR workflow. The first case study will look at quantifying T-cell activation response to checkpoint inhibitors, and then the second case study will look at the effects of checkpoint inhibitors on inflammation. Advanced flow cytometry is used in a wide array of biological applications and has evolved into a technique for rapid analysis of DNA content, cellular marker expression, and electronic cell sorting. It is used extensively for monitoring phenotype expression, drug transport, cell cycle proliferation, and apoptosis. Advanced flow cytometry has much higher throughput compared to more traditional flow cytometry methods, which has greatly increased its use as a tool for screening and profiling studies. The importance and contributions of advanced flow cytometry in drug discovery continues to build, particularly in the field of immuno-oncology. So this is the iQue3 platform. The iQue3 is an advanced flow cytometer that is capable of reading from either 96 or 384 well plates at high speed. It typically takes about 10 minutes to read a full 96 well plate or about half an hour to run a full 384 well plate. As well as this being beneficial for screening applications, the high throughput also facilitates replication for generation of more robust data. It has a total of 15 detection channels, which means that a vast quantity of information can be collected in a single run. Sartorius have a large portfolio of optimized reagents and assay kits, each of which are designed to minimize the amount of precious sample required, with the cell and supernatant volumes required to run most of the kits ranging from about 5 to 10 microliters. The iQue3 system comes with built-in foresight software, which allows for fast time to results as the graphs auto-populate during cell sampling. The software is very easy to use, which when combined with the preset gating and analysis templates provided with the reagent kits, drastically simplifies and speeds up the data analysis and presentation. So this will be exampled in some of the data that I'm going to show today, all of which has been generated in Foresight. And then here are some examples from the suite of kits compatible with iQue3. So these are applicable to a range of different applications, namely within the areas of immuno-oncology, antibody discovery, and foundational cell functions. The iQue allows a user to simultaneously test T-cell phenotype and secreted protein concentrations in each well of a 96 or 384 well plate. So this gives insight into the status of multiple cell types within a complex cell environment, for example, through the quantification of CD3 positive lymphocyte populations, or through live and dead cell readouts, and through cell marker expression. The Q beads that are also included in the wells also allow for the simultaneous capture and quantification of multiple secreted protein or cytokine readouts. So for example, TNF-alpha and granzyme B. The synapse between T cells and APCs, such as dendritic cells, is a key site of immune regulation. And in order for the regulatory information to be transferred across this synapse, there are three main types of signal used. The first is antigen-specific recognition, which involves presentation of antigens by the MHC molecules on the surface of the APC. These antigens are then recognized as foreign by the TCR on the molecules on the surface of the T cell, which triggers T cell activation. Following this antigen recognition is secondary signaling by co-stimulatory or co-inhibitory molecules called immune checkpoints, which act to either increase or decrease the T cell response. And then finally, the immune response is propagated by chemokines and cytokines that are produced and recognized by both the T cell and the APC. So the assay that we're going to be discussing today is used to measure the effect of checkpoint inhibitor drugs that act to specifically block the co-inhibitory immune checkpoint molecules. So to do this, we're going to measure the downstream effects of the immune checkpoint interactions by quantifying T cell proliferation, marker expression, and cytokine release, all of which can be reliable indicators of T cell activation. So in physiological conditions, the function of inhibitory checkpoint molecules is to induce a decrease in T cell activation to prevent an overactivation of the immune response, as this can lead to an attack on healthy body cells. Checkpoint inhibitor therapeutics are monoclonal antibodies that bind to and block this interaction. Some examples include anti-CTLA-4 antibody ipilimumab and anti-PD-1 antibodies such as nivolumab and pembrolizumab. These are both targets found on the surface of T cells. Similarly, there are drugs that target their partner proteins found on dendritic cells, such as anti-PD-L1 antibody durvalumab. In blocking these interactions, checkpoint inhibitor drugs act to take the brake off of T cells, leading to enhancement of their activation response, which in turn should lead to an increase in their ability to kill tumor cells. Often, several of these drugs with different target proteins are used in combination to induce a synergistic effect on T cell response. The mixed lymphocyte reaction is a way of mimicking the interaction between T cells and APCs in vitro. It involves co-culturing immune cells from two different donors to trigger the allogenic T cell activation response. There are two different types of MLR assay that I'm going to show example data from today, and these are called one-way and two-way MLR. So the one-way MLR involves taking CD4 positive T cells from one donor and combining them with dendritic cells from another donor. The cells are co-cultured for around 3 to 7 days, during which time the DCs stimulate the activation and proliferation of the T cells. Two-way MLR is very similar, except that rather than co-culturing isolated immune cell populations, instead PBMCs from two donors are combined at a 1-to-1 ratio. And this creates a two-way activation response with the DCs from each donor activating the T cells from the other donor. So the result is activation and proliferation of both donor 1 and donor 2 T cells. A key contributor to the activation response in MLR is the presence of mismatching HLA antigens on the surface of the cells from each donor. So for example, if the two donors have very similar HLA profiles, the allogenic response won't be triggered and T cells won't be activated by the DCs. So this is why in the experiments I'm going to show you, we either selected donors based on their HLA profile, or we compared the response across multiple donor pairs. So I'm now going to give a brief overview of the iQue MLR assay workflow. First, we combine the immune cells from the two donors with treatments in the cell culture plates. Then if we're going to perform downstream analysis of the cell subtypes, we can choose to label our T cells or PBMCs with the iQue proliferation and encoding dye. This is either included as part of the T cell activation kit, which I'm going to give some more detail on in a minute, or it's available as a standalone reagent. Samples are then transferred to V-bottom plates for labeling. And because of the fact that each kit only requires a 10 microliter sample volume, multiple samples can be taken from the single assay plate for analysis at multiple time points or using multiple kits. The iQue kits that we have validated this assay using include the human T cell activation kit, which measures proliferation, general cell phenotyping with CD3, 4, and 8, as well as activation markers and cytokines. We also performed analysis using QBead-only kits for measurement of other cytokines and chemokines, for example, the information panel kit, or we used customized QBead screening panels. The plates are then washed once and then run on the iQue. So now I'm going to give some more detail on the T cell activation kit, as this is the kit that was used to generate most of the data that I'm going to show you today. So as I introduced in the previous slide, this kit enables measurement of both cell subset and cytokine concentrations all on a single well. The kit contains an antibody cocktail, a viability dye, a proliferation and encoding dye, and QBeads for cytokine detection. The kit also comes with a preset gating template, which can be imported into the iQue built-in Foresight software, and this means that readouts for the T cell activation can be generated automatically. So now I'm just going to give a quick overview of the preset gating template that's provided with the T cell activation kit. So the first plot is used to separate the cells from the cytokine capture beads. And then within the cell gate, we gate for the single cell population and then gate to separate the live and dead cells. And then it would be at this point, if we'd included the optional proliferation encoding dye, that we could, for example, separate green labeled T cells from the unlabeled DCs. Then within that gate, we can gate for the CD3 population, then the CD4 and CD8s, and then finally within those gates, we'll gate for the activation markers, which in this kit are CD69, CD25, and HLA-DR, which respectively give an indication of early, mid, and late T cell activation. To help to double check and sort of fine tune the positioning of the gates, the positive and negative control wells can be defined, and then these will automatically be overlaid by the software to help to distinguish your positive populations. The gating is also carried through from the bead gate, so through from the single beads, and then finally to separate each of the bead types based on their unique fluorescence signature, and the signal for the secondary antibody fluorescence on each bead type can be used to quantify each cytokine. So before we move into the case studies, I'm going to show a data set that exemplifies the importance of the donor HLA haplotypes on the level of T cell activation, as I started to introduce earlier. So in this experiment, we took dendritic cells from one donor and co-cultured them with T cells from 2 different donors, and then after 5 days, analyzed cells using the T cell activation kit. So the T cell donors we used were selected based on the level of mismatch between their HLA alleles and the DCs. So one of the T cell donors was similar in HLA profile to the DC donor. This was the low mismatch pair, whilst the other T cell donor had a largely different HLA profile, and this was the high mismatch pair. The cells were co-cultured at a range of T cell to DC ratios, and then the T cells in monoculture with and without CD3, CD28 dynabeads were used for positive and negative controls. So at the top is a plate view diagram taken from Foresight, which shows plots from individual wells containing the low and high mismatch co-cultures at each T to DC ratio. So expression of activation marker CD25 is plotted along the x-axis, and the side scatter or granularity is on the y-axis. And it's clear from this diagram that as the T cell to DC ratio decreases from 9:1 to 3:1, which corresponds to an increase in the number of DCs per well, the degree of T cell activation in the high mismatch co-culture increases much more drastically than in the low mismatch co-culture. And then this same data is just summarized in the graph in the bottom left here, where we can see that the low mismatch at the 3:1 T to DC ratio, there was only 8% CD25 expression compared to 34% with the high mismatch. Interestingly, the CD25 expression in the low T cell only control was higher than in the MLR, perhaps indicating a level of non-APC related autoactivation by this donor. Overall, a very similar profile was also seen for the T cell proliferation, and together these data show how important the HLA mismatch is for the allogenic T cell activation in MLR. So I'm now going to discuss two case studies that use the MLR assay to quantify the response to checkpoint inhibitors. So the first case study will look at quantifying T cell activation and will be explored through two different experiments. So the first is a simple one-way MLR assay looking at the effects of pembrolizumab, which is an anti-PD-1 checkpoint inhibitor antibody, on activation, marker expression, and cytokine release. And then the second experiment is the two-way MLR assay, investigating the synergistic effects of using checkpoint inhibitors in combination. And then the second case study will look at how checkpoint inhibitors can impact the production of inflammatory cytokines. So moving into that first activation experiment. So here we selected a pair of donors with a high level of mismatch in their HLA, and then the activation response was measured using the iQue T cell activation kit. So the contour plot on the right shows that after 6 days in co-culture with the DCs, the CD4 positive T cells had nearly 3 times higher expression of CD25 when compared to the T cells in monoculture in the plot on the left. We then added pembrolizumab to the co-culture and saw that a further concentration-dependent increase in CD25 expression up to a maximum of about 75% was observed. This drug-induced increase in the T cell activation is not as pronounced as the threefold increase we initially saw due to the addition of DCs, but this might not be surprising and may actually reflect the clinical situation in that the aim of the checkpoint inhibitor is to delicately tip the balance towards enhanced T cell activation rather than to overactivate the T cell response. The two cytokines detected by the T cell activation kit are TNF alpha and interferon gamma. And so we took samples on days 2 and day 6 and analyzed the concentrations of both these cytokines. Both cytokines increased in response to pembrolizumab, but it was clear that each showed a difference in how it was released over time. So the TNF alpha release was high on day 2, but levels diminished by day 6, whereas the interferon gamma release was low on day 2, but increased to high levels by day 6. But despite these temporal differences, the EC50 values for the release of these cytokines in the response to pembrolizumab were very similar, with a value of 0.54 micrograms per mil for the day 2 TNF alpha release, compared to 0.44 micrograms per mil for the day 6 interferon gamma release. So this highlights why it could be useful to analyze at multiple time points to really build up a fuller picture of the drug response. So now I'm going to show the activation experiment looking at the effect of using checkpoint inhibitors in combination. So this experiment was a two-way MLR assay, which was comprised of a co-culture of PBMCs from 2 different donors, and the PBMCs were incubated for 3 days with 10 different combinations of antibodies. So these either included the checkpoint inhibitors on their own or in combination with other checkpoint inhibitors. And the treatments were compared to 2 negative controls, marked here as C, which is the absence of any antibody, and M, which is MMA, and then PMA, which is marked here as P, was included as a positive control. And then this heat map just shows the TNF alpha release in nanograms per mil in 2 replicate rows of PBMCs, and the graph here summarizes the same data. And it was clear that the most striking effects on TNF alpha release were observed when the anti-CTLA4 antibody was added. And in particular, there was a clear increase in response when this antibody was added in combination with other checkpoint inhibitors. And we saw this up to a maximum of an 8-fold increase when both the anti-CD73 and the anti-CTLA4 antibodies were added in combination. We also looked at the interferon gamma release, which was also greatest in the wells containing the anti-CTLA4 antibody. However, the effects of including other checkpoint inhibitors in combination with this were minimal when you compare to what we saw with the TNF alpha. So this just really highlights why it can be useful to look at a few different cytokines in multiplex to gather a full characterization profile for a drug's effect. If I also show you a fuller version of the heat map that was produced in this experiment, you can see that we actually tested these same treatments with a total of 3 PBMC donor pairs. And we saw that whilst the cytokine release profiles for donor pairs 1 and 3 were very similar, there was minimal drug-induced response in comparison with donor 2. So this highlights the importance of testing drug activity across a range of different donors. Finally, I'm going to look at the impact of checkpoint inhibitors in the context of the inflammatory response. So very common side effects of treatment with checkpoint inhibitors are due to an overactivation of T cells causing an attack on healthy body cells, which can result in adverse inflammatory response. This inflammation is associated with high production of inflammatory cytokines, such as CCL2 and interferon gamma. And although these cytokines are produced as part of a normal activation response, when the production is upregulated, it can start to propagate the inflammatory state. So to investigate the effects of pembrolizumab on inflammation, we co-cultured DCs and T cells in a one-way MLR assay, and then analyzed the supernatant cytokine concentrations at days 2 and 6 using the iQue Human Inflammation Panel kit. So this kit measures the concentrations of 7 pro-inflammatory cytokines and chemokines in multiplex. And these plots show the preset gating template that's provided with the kit, which similarly to the T cell activation kit, helps us to separate our bead populations and to quantify each secreted protein concentration. So here I've got a few examples of the data generated for cytokine release in response to pembrolizumab. So on the left is an example of the raw IL-2 concentration data on days 2 and 6, displayed in heat map form, which clearly shows that there was high level and concentration dependent IL-2 production on day 2, which has diminished by day 6. The same data is then summarized as a concentration response curve in the middle. And then there's also a couple of other examples with CCL2 and CCL3, both of which increased from days 2 to day 6, but also showed pembrolizumab concentration dependence on day 6 with very similar EC50 values of 0.98 and 1.01 micrograms per mil. And I haven't shown the data here, but the levels of interferon gamma, IL-6 and CXCL10 also increased with the pembrolizumab concentration. So overall, this all supported the idea that checkpoint inhibitors can increase the inflammatory response. Finally, we wanted to see if including a corticosteroid could impact the level of inflammatory cytokine release in MLR, as these were often the first-line treatment for adverse inflammatory events associated with checkpoint inhibitor therapy. So here we have data showing the interferon gamma release over 3 time points with either T cells only, T cells co-cultured with DCs in MLR, and MLR with a single concentration of pembrolizumab added. And the interferon gamma release in each condition was compared in the presence and absence of corticosteroid dexamethasone. And it was interesting to see that on days 4 and 7, both in the presence and absence of pembrolizumab, we saw a decrease in the concentration of interferon gamma that was produced when the dexamethasone was added. And this same effect was also seen across several of the other inflammatory cytokines in the kit. However, it is worth noting that whilst the concentrations of most of the inflammatory cytokines in the kit decreased in the presence of dexamethasone, the concentrations of 2 of the cytokines, CCL2 and CL10, actually increased when the dexamethasone was added. So whilst it's clear that the corticosteroid is having an impact on the inflammatory cytokine release in MLR, it would definitely take further investigation to determine what the summative impact of these effects would be in vivo. So to summarize, the iQue Advanced Flow Cytometry Platform and compatible reagents provide a simple workflow for quantifying T cell response to checkpoint inhibitor drugs. The preset gating templates provided with the kits, alongside user-friendly data presentation and analysis features, mean the readouts can be generated quickly and easily. And then combining this with the high throughput of the iQue creates a powerful tool with applications in biological research and potential to enhance drug discovery processes. For further information, please see these following links, which will take you to iQue MLR webpage and application note. And then I've also included links to the T-cell activation kit and the QBead information panel kits, web pages and protocols for further information on the reagents used. Thanks very much for listening. I hope you enjoyed the webinar, and I look forward to answering any questions you might have.
Thank you for that, Kirsty. That was a great presentation. Before we continue to the questions, where Kirsty will be joined by Nicola Bevan, Manager, Cell Imaging Applications, Bioanalytics Product Development at Sartorius, I'd just like to remind everyone that this webinar will also be available to watch on demand on the DDW website, www.ddw-online.com. Okay, over to you, Kirsty and Nicola, for questions.
Thanks very much. So yeah, just going to start with this question here. So which alleles were considered for HLA mismatch testing and which assay was used? So the alleles that we looked at were each of the two HLA-A, B, C, and DR alleles, so 8 alleles in total. And in terms of how this was done, actually, we just had the information provided to us by the cell supplier. So this was primarily Stem Cell Technologies. And so we were just given a list of donors and their HLA profiles and then selected what was going to suit us for each assay. And then another question here, what was the DC to T cell ratio in the one-way MLR? So aside from that one assay where we had a look at different DC to T cell ratios, we just stuck to the 3:1 T cell to DC ratio throughout the rest of the one-way MLR assays, as we found that that had the optimal activation response. So another question here, what are the advantages or disadvantages of using one-way compared to two-way MLR? So the main advantage of the one-way model is that you get sort of simple unidirectional activation of the T cells without much background signal, which would be the main disadvantage of the two-way model, as this gives a more sort of crude measurement of the activation and cytokine release due to the presence of multiple cell types from each donor. The disadvantage of the one-way MLR is that it requires isolated immune cell populations, which is generally going to be either more time-consuming or more expensive to acquire compared to straight PBMC samples that are required for the two-way MLR. So another question, in two-way MLR, could you label in a way that allows proliferation of T cells from each donor to be distinguished? So yes, you could. So there are 3 different proliferation encoding dye kits in the iQue portfolio, and each that fit in one of the violet, blue, and red channels. The reason we have only used the blue-green dye in all of these experiments is that it's compatible with the T-cell activation kit in terms of the markers and fluorophores used. However, if you wanted to sort of look at your own bespoke antibody panel and alongside some cytokines, then you could easily look at multiple proliferation dyes and look at the proliferation of T cells from each donor. So another question, how does this assay compare to traditional MLR assays? So I think one of the sort of main differences with this assay compared to the more traditional techniques is that you're looking at all the parameters in a single well. So traditionally, you'd need to use, for example, a separate assay for marker expression. So for example, with traditional flow, and then also proliferation, and then probably cytokine release, you'd use a separate ELISA. But looking at everything in a single well in this format, it drastically speeds up both assay time and your data analysis, means you need less sample volume, and also it should eliminate any variation due to sort of measuring outputs from different cell populations.
Okay, thank you for that. And thank you to everyone who submitted questions. I'd just like to say thank you to Kirsty and Sartorius for such a detailed and useful presentation. I'm sure DDW listeners have taken away some valuable information there. If you'd like to listen to the webinar again, it will be available on demand at www.ddw-online.com. You can now view Drug Discovery World content online, on your mobile or tablet, or via the new DDW app, which can be downloaded via the App Store or Google Play. Thank you for listening.